Hi everybody! Today we'll be talking about how to properly make a gram stain slide. At the end of this training video, medical laboratory students will be able to properly perform gram stains using crystal violet, iodine, ethanol or acetone, and safranin to obtain accurate color representation of bacteria. Additionally, medical laboratory students will be able to identify how gram stain reagents interact with both gram-positive and gram-negative bacteria based on the biochemical and structural differences. Before we begin, it's important to note that there are two main types of gram stains, the direct and the colony. The direct is done by gram staining the direct sample that is given to the lab, and the colony is done by growing the sample out on a plate. This is typically reported within 24 to 48 hours of receiving the sample. For the purpose of this video, we will be performing a colony gram stain to perform quality control. Now let's go over the needed materials. We'll need sterile swabs, a biohazard bag, inoculation loop, loop sterilizer, a rack with tube of sterile saline, paper towels, glass slides, a wax and graphite pencil, plate with the organism needed grown on it, a sink with running water, staining tray, and staining reagents which include crystal violet, iodine, ethanol or acetone, and safranin. Additionally, you will need a cup to hold water in the sink, hemostats, and a microscope, and optionally a heat block. The first step to making a gram stain slide is to first grab one of the slides and a graphite pencil. Label the slide on the frosted portion of the glass with the technician's initial, date, and sample information. In this case, we will be doing a QC slide. After, grab the wax pencil and draw a circle on the clear portion of the slide. Take a tube filled with saline solution and put a sterilized swab in it. After the swab is damp, take the tip of the swab and place it on the center of the circle on the slide. After doing this, sterilize the loop. The loop should be orange immediately after taking it out if it is properly sterilized. Obtain a plate with the needed organism grown on it. This is preferably done with an SBA plate. Take the cool sterilized loop and place it on a colony. In this example, we are grabbing the gram-negative organism on the plate. Place the loop with the colony on the slide with the saline. Spread the organism within the circle, making it evenly distributed. After mixing the organism in the saline, re-sterilize the loop and repeat the previous steps to place the gram-positive organism on the slide. During this process, make sure not to grab too much organism. If this occurs, it will make the slide harder to stain and therefore harder to read later on underneath the microscope. After this is all done, the slide will need to dry. This can be achieved by letting the slide air dry or setting on a heat block face up as seen. After the organism is dried on the slide, it will need to be heat fixed. This is done by taking the slide with hemostats and placing it above the incinerator for approximately 3 to 5 seconds. Now it's time to stain the slide. With water running in a cup, place the slide in the staining bath. Take the crystal violet stain and pour it on the slide, completely covering it. Wait for 30 seconds. Here we have a gram-positive cell surface. On the top we have peptidoglycan layers as well as teichoic acid and lipoteichoic acid. These make up the cell wall. Underneath we have the plasma membrane containing a phospholipid bilayer with integral proteins. During the first step of staining, the crystal violet penetrates these layers, completely staining them. Now let's look at our gram-negative cell surface. There are several differences that we see comparing to the gram-positive cells. On the top we have an outer membrane which is composed of a phospholipid bilayer. In this layer we also have proteins and lipopolysaccharide, also abbreviated as LPS. The LPS is combined of O-polysaccharide and lipid A. Underneath we have a much smaller peptidoglycan layer. Those two layers combined make up our gram-negative cell wall. Underneath we have an additional phospholipid bilayer that is referred to as our plasma membrane. This layer is the same as our gram-positive cells. Similarly to our gram-positive cell, the crystal violet stain penetrates the gram-negative cell the same way, by completely staining each layer. After 30 seconds, take hemostats and tap the crystal violet off into the staining tray. Take the slide and rinse off the remaining crystal violet stain in the water-filled cup. Take the iodine stain and pour it on the slide, completely covering it. Wait for 1 minute. The iodine intensifies the crystal violet stain. It combines with the CVI complex trapped in the gram-positive cell's peptidoglycan layer. The iodine also acts similarly with a gram-negative cell's peptidoglycan layer. However, because the peptidoglycan layer is much smaller compared to our gram-positive cells, it's less effective in holding the crystal violet. We'll see this later on with the other stains. After the iodine is set, take the slide with the hemostats and tap off the stain in the staining tray. Take the slide and rinse off most of the iodine in the water-filled cup with a little bit of iodine left on. Take the ethanol or acetone and cover the slide over the sink, rocking the slide for 8 to 10 seconds. Immediately after, rinse all the ethanol or acetone off the slide and set it back in the staining tray and completely cover it in the safranin stain. Let the safranin set for 30 seconds. During the ethanol or acetone stain, the phospholipid layer in both the gram-positive and gram-negative cell will slightly be dissolved. In the gram-negative cell, the ethanol or acetone will wash the CVI complex from the cell wall, completely washing away the crystal violet stain. After adding the safranin, the gram-positive cell will hold some of the stain. However, because the cells are already holding the crystal violet stain, the pink to red color of the safranin is not seen underneath the microscope. The gram-negative, on the other hand, has been washed of the crystal violet stain and will hold the safranin. After the 30 seconds are done, take the slide with hemostats and tap off the stain in the staining tray. Rinse off the excess stain in the water-filled cup, making sure that all the stain is removed. Place the slide on a paper towel and carefully pat it down. After, make sure to empty the cup in the staining tray to clear the line of all stain and then turn off the water. And there you have it, guys, the proper way to make a gram stain slide. Big thanks to my group members Leslie and Liza for helping me make this video. Thank y'all for watching.